Hello everyone. This is Chapter 8, Part 1 of Unit 3. Already we have completed 2 units. In this unit, we have to see that is cell structure and functions. This unit also includes 3 chapters. This is Chapter 8, Part 1. In this chapter, we have to see that is cell, the unit of life. And in this part, we have to discuss a part of this, that is cell theory and overall view of cell, that is detailed structure of prokaryotic cell and how does it differ from a eukaryotic cell. This is Part 1, Chapter 8. This is Unit 3, Cell Structure and Function. This unit consists of 3 chapters, Chapter number 8, Chapter number 9 and Chapter number 10. Here we have to deal Chapter number 8 first. You see, for this unit, that is 𝕍𝕆𝕋𝕍𝔼 is the name who has done outstanding work. That is the figure of 𝕍𝕆𝕋𝕍𝔼. That is, he is famous for, that is, discovery of triple helical structure of collagen protein. He published his work in Nature in 1954, and Ramchandran plot is very famous work related to the protein structure. He received his PhD from Cambridge University, and his work is outstanding in field of structural biology. Now the chapter Cell, the unit of life, Chapter 8, we have to deal part first of this. You are familiar, children, that all organisms are composed of cells, and cell is the basic unit of life. You see, the organism may be unicellular, made up of only one cell, or made up of many cells, multicellular organism. Even if it is made up of one cell, all functions of life are being done by single cell. Unicellular organism, they are having independent existence and they can perform all essential functions of life. You see, anything less than a complete structure of a cell doesn't ensure independent existence. Therefore, cell is known as structural and functional unit of cell. We have to see in time to come what actually we mean by cell. The first cell was seen by, that is, Antony von Leeuwenhoek, and he described a live cell. Robert Brown, that is, he discovered the nucleus inside cell, and later on the invention of microscope and its improvement leading to the, that is, electron microscope, that has revealed detailed structure of cell and different organelles of cell. Now we have to see what we mean by cell theory. You see, cell theory, how to understand. Now we mean by cell, we mean that is, this is a cell. This is a plasma membrane, outermost layer. That is, this is the nucleus. This is the nuclear membrane. Inside the nucleus, we have, that is, genetic material. And here we have this one is the cytoplasm. Such a structure actually is known as a cell. This is the unit of life. You see, that is, in 1838, Schilden, a German botanist he had been, he examined number of plant cells and he observed that all plant cells are composed of different type of cell and which make the tissue, right? At the same time, that is, T. Swan, he was a British zoologist. He studied different type of animal cell. Schilden is, that is, related to plant cell. Here the Swan studied different type of animal cell, and he reported that the cell outermost layer is a thin outer layer that is known as a plasma membrane. This is the plasma membrane, which is the outermost thin layer. He also concluded that is the plant tissue, if this is a plant tissue, then in case of plant tissue, outside this plasma membrane, a thick, that is, non-elastic layer is also found, which is known as, that is, cell wall. He told presence of cell wall is a unique character of plant cell. On the basis of this, Swan proposed hypothesis that the bodies of animal and plants are composed of cells and cell products, right? Then little later, Schilden and Swan together, they formulated cell theory. What we mean by cell theory, that is, however, that didn't explain how the cell arises, how new cells are formed, but addition was being made by Rudolf Virchow in 1855. He for the first time told that the cells are formed from pre-existing cell. By division of the pre-existing cell, new cells are formed. In his own language, he called this statement is omnis cellula e cellula. Very important statement. What we mean by this, that is the new cells are formed from pre-existing one, and he modified hypothesis of Sheldon and Swan to cell theory, and he gave the final shape. Okay? That is omnis cellula e cellula. It is known as, that is, cell lineage, and a number of other modifications were carried out later on, and the modern cell theory is known as, that is, principle, or that is cell doctrine. Is it right? Now we have to say some objections related to cell theory. Again I have to repeat, what in by cell now? That the cell is having outermost, that is plasma membrane. It should have a well-defined, that is nucleus, outside the nucleus, and that is in between nuclear membrane and plasma membrane, that is cytoplasm is there, that is cytoplasm, right? But you see that the organism which do not have such type of structure, that will be objection to cell theory. Virus do not have cellular machinery. In virus we do not have cytoplasm. That's why it is an objection to the cell theory. Bacteria and blue-green algae, they do not have well-defined nucleus. That's why we cannot call them as cell. Certain fungi like Rhizopus, the hyphae is composed of, that is, this is the hyphae, it is having, that is, more than one nucleus. These are sinocytic one. Multinucleated mass, that's why this is also an objection. As tabularia, a unicellular marine algae, this is made up of only single cell, but this single cell is having differentiated plant body. This is an objection. Red blood cells and the sieve tube, they continue to live without nucleus, and that is other vital organ. That how they can survive, because they are not cell. This is objection to cell theory. Is it right? Come to the now next point. We have to see overview of cell. That is, first you have to see cell size and shape. You are seeing different type of, this is a typical eukaryotic cell. The size is in between 10 to 20 micrometer. This is a typical bacteria, 1 to 2 micrometer. That is 𝕆𝕋ℍ𝔼𝕆-like organism, mycoplasma. The size is, that is 0.1 micromu, and the viruses, they will be even smaller, 0.02 to 0.2 micrometer. So size varies. Cell differs greatly in size, shape and activities. For example, the same thing, mycoplasma. Mycoplasma is the smallest cell, that is are only about, that is 0.3 micrometer in size, while the bacteria, they range 3 to 5 micrometer. Largest isolated single cell is egg of ostrich. Among multicellular organism, human red blood cells important, that is come in examination. The size of RBC, it is having about 7 micrometer in diameter. Nerve cells are some of the longest cells. They are also very great in shape. That is, they may be disc-like, they may be polygonal, they may be columnar, they may be cuboidal, they may be thread-like, or even they may be irregular one. Shape of the cell, it may vary with the functions they perform. The structure of the cell, shape of the cell is very much correlated with the function performed by cell. You can see some of the shapes are given. That is, this is red blood cells, that is round end, that is biconcave. This is white blood cell, this is amoeboid one, columnar, these are the long and narrow. This is the nerve cell which is branched and the long one. This is trachea found in plant cell. This is elongated one, and mesophyll cells, these are the round one. Different shape of the cells you have seen right now. Now we have to see a typical plant cell. You see onion peel. We can prepare a slide. It is having a distinct cell wall as the outermost boundary. Inside each cell there is a dense membrane structure, that is called nucleus. It contains, that is nuclear material, that is chromosome, which is the genetic material of the cell. And prokaryotic cell, if I am going to make that, this is the prokaryotic cell, that is here, this is the plasma membrane, and that may be structure outside the plasma membrane. And here, this is the, that is so-called nuclear, that is material. It doesn't have a well-defined nucleus. This is the chromatin what we have here. It doesn't have well-defined nucleus, while in case of eukaryotic cell, well-defined nucleus is there. In this case, in prokaryotic cell, the cytoplasmic organelles are absent. Only, that is 70S, that is ribosome, is found here in case of prokaryotic cell, while in case of eukaryotic cell, we have different type of, that is cytoplasmic, that is organelles, and these cytoplasmic organelles are bounded by membrane. These are membrane bounded cytoplasmic organelles. Here the cytoplasm, that is the main arena of cellular activities. Various chemical reactions occurs in it to keep the cell, that is, in living state. We have to see detail of, that is, prokaryotic cell. Now, prokaryotic cell, you see what we mean by prokaryotic cell. You know, in 5 kingdom classification I told you that was kingdom, that is Monera, and kingdom Monera, it is having the prokaryotic cell. Prokaryotic cell, you know, the cell doesn't have well-defined nucleus. Prokaryotic cell are represented by bacteria, blue-green algae, mycoplasma, and that is PPLO. You see, these are usually smaller in size. They multiply more rapidly as compared to the eukaryotic cell. Important characteristic this one is. They may vary great in shape and size. The 4 basic shape of the bacteria you are familiar. That is, this is Bacillus, Coccus, Vibrio, and that is Spirillum, spiral shaped. Organization of the prokaryotic cell, definitely fundamentally it is similar through, that is, the prokaryotic cell. They exhibit a wide variety of shape and functions. Lot of variety of prokaryotic cell, but the fundamental, that is, structure and function is almost same of all the prokaryotic cell. We have to see some basic things now. That is cell envelope. You see, let this is the, that is cell. Here we have prokaryotic cell. Let this is the plasma membrane. Outside the plasma membrane, this one is the, that is, cell wall, and outside the cell wall there is one more structure known as, that is, all these 3 together, that is known as glycocalyx. The outermost one is known as, that is, glycocalyx. The second one is known as, that is, cell wall, and the innermost one, that is known as, that is, plasma membrane. All these 3 together, they will make the, that is, cell envelope. Most of the prokaryotic cell, that is especially the bacterial cell, they have chemical complex cell envelope. As I told you right now, the cell envelope, it consists of tightly bound 3 layers structures, and these 3 layer structure, can you recall it now? The outermost one is, that is, glycocalyx, followed by, that is, cell wall, and it is followed by, that is, plasma membrane, that is, envelope. Now you can see the structure of bacteria here. A typical bacteria I had told you here, that is, you see, it doesn't have well-defined nucleus. This is the nucleus. Here we have incipient nucleus, or also known as, that is also called xenophore, right? This is the cell wall. Here we have, outside the cell wall we have, that is, capsule. That is, in this case, that is outside the main so-called nucleus, some DNA is found in cytoplasm also, that is known as plasmid. This is cytoplasmic, that is, membrane. I told you 70s ribosomes are found. This is the only cytoplasmic organelles. These are some other substances which are found in bacterial cell. That is attachment. These are the pili which help, that is, attachment to any other that is surface, and this is the outer membrane found only in case of gram-negative bacteria. We have to see what in by gram-negative and gram-positive bacteria. Usually flagella is found in case of gram-negative bacteria more frequently, right? Now you see, each layer perform distinct function of this cell envelope, but they act together as a single protective unit. Is it? The main function of all this cell envelope is protection. Bacteria can be classified into 2 groups. The detail we don't have here in course, on the basis of difference of cell envelope in the manner in which they respond to the staining procedure, that is known as crystal, that is, violet stain is used, as that was used for the first time by scientist Gram. That's why these are known as Gram stain. The bacteria which retain Gram stain are known as gram-positive one. These are called gram-positive, and in stain, the stain will be washed out. That will be known as, that is, gram-negative one. So you see the detail of this now. Glycocalyx differs in composition and thickness among different bacteria. This is the outermost. It could be loose sheath called slime layer, or it may be a thick and tough one known as capsule. Capsule is usually found in, that is, gram-negative bacteria. Cell wall, that is, cell wall, it means the shape of the cell. It provides strong structural support, and it prevents the bacteria from bursting or collapsing. This layer is rigid, very important point, and made up of peptidoglycan, that is mucopolysaccharides. This is mucopeptides, peptidoglycan. Number of, that is antibiotics. Penicillin inhibits cross-linking of peptidoglycan strands. That's why that is penicillin is used to kill bacteria, because penicillin will not allow formation of cell wall and the bacteria will die. That is peptidoglycan will not be formed here. Okay. Then we have to say related to cell wall some, that is modifications are there. One is that is flagella. Bacteria cell, it may be motile or non-motile. The cell which are motile, they have flagella. That is, this is the thin filamentous structure. You see, if this is the bacteria we have, that is, this is the cell wall here. This is plasma membrane. This is the structure inside the body of the bacteria. Now this is a hook-like structure, and in hook-like structure we have, that is, this is the flagellum. So here this structure is called flagella. Bacteria show a range and number of flagella. It may be one, it may be many more, it may be throughout the surface, or it may be only at one pole. Bacteria phloem is made up of, that is, three parts. One is filament, one is hook, one is basal body. This is filament, this is hook, and this one is the basal body. The filament is longest portion, and it extends from the cell help, that is outer side, and it help in, that is, movement of the bacteria. Pili and fimbriae, you see, pili, these are found, these are the small projections here, pili on the surface of bacteria, and with the help of this pili, that is conjugation, you have to study in time to come. Conjugation, that is, will take place, through which, that is, plasmid will be transferred to other, that is, bacterium. In the same way, that is fimbriae, these are many more like this, fimbriae structures, and these fimbriae, these are also surface structure found in bacteria, but they do not play any role in motility. The pili are elongated tubular structure, as I told you. They help in conjugation, and the fimbriae, they help, that is, in attachment to certain surface. Either this is rock or to some other host tissue. The bacteria will be attached with the help of this fimbriae. Is it right? Now, next we have to see, that is plasma membrane. Plasma membrane, see, again you have to see, that I'm going to make here, that this is the bacteria. You see, that is, this was the outermost one, glycocalyx. That was the second layer. That is the cell wall. This is the cell wall here. That one is, that is, the outermost one is glycocalyx. While surrounding this cytoplasm, that is, this is the outermost layer, which is known as, that is, plasma membrane. You know, all the three together, they make the cell envelope. So this plasma membrane, this is selective permeable, and it interact with the outer world. What will go inside, what will go from outer side, that is inside, or what will go from inside to outer side, that is being controlled by plasma membrane. It is special membranous structure. What is important, that is this plasma membrane that get invaginate inside, and that make special membranous structure. These membranous structure are known as mesosomes. Very important, mesosomes. Mesosomes, these are made up of, by invagination of plasma membrane, extension of plasma membrane into the cell, mesosomes are formed, and these may be in the form of vesicles, in the form of tubules, or in case of lamellae. Very important point here. What the function of these mesosomes? They help in cell wall formation. When the bacteria, single bacteria, will go binary fission, and here the cell wall is to be formed, the cell wall formation, that will be facilitated by, that is, mesosomes. DNA replication and distribution, that is to the daughter cells. They also help in respiration. That is, here they have, that is, respiratory enzymes, and that's why they help in respiration and secretion process. To increase surface area of the plasma membrane, because here the membrane surface will be increased, and therefore the area for enzymatic activity will also be increased. Right? An important point we have here, that is, in mycoplasma, cell wall is absent. It is very important one. That's why, can you recall, when the cell wall is absent, the mycoplasma can be treated by penicillin? No, because penicillin will not affect the mycoplasma, because the cell wall is absent in case of mycoplasma. Is it right? Okay. So you have to see the next one now. Certain cytoplasmic organelles, we have to see, as I told in the beginning, that is in case of a prokaryotic cell, we have this is the, that is nucleus so-called, which doesn't have nuclear membrane, and this one is the cytoplasm. In cytoplasm, we do not have cytoplasmic organelles, which are membrane bounded one. As I told you, very important one. Only an organelle is there, which is known as ribosome, and this ribosome is of 70S type. It is made up of 2 units, the smaller unit of 30S and the larger unit is of 50S. This is smaller one, this is the larger one. This is only the cytoplasmic organelle, and the ribosome, you know, it doesn't have any membrane. So ribosome, you see, no organelle that is found, as are found in case of eukaryotic cell. These organelles are absent here in this prokaryotic cell. Ribosomes in prokaryote, come on, in prokaryote ribosomes are associated with plasma membrane. These, the size will be 15 nanometer to 20 nanometer in size. As I told you, made up of 2 subunits, 50S and the 30S, and that will make the 70S prokaryotic ribosome. Ribosome, you know, it is the site of protein synthesis. Several ribosomes may be attached to single messenger RNA. That this is the messenger RNA. That is, at the time of protein synthesis, so many ribosomes will be attached to this, and in that case, this structure is known as polyribosome or polysome. Is it? And here, the ribosome of a polysome translation of messenger RNA will take place, and the protein will be synthesized here. In some prokaryotic cell, which used to do photosynthesis like cyanobacteria, you know, cyanobacteria were known as blue-green algae. They are green in color. They do photosynthesis. The first photosynthesis on earth in which the oxygen was being released, you know, I told you earlier, that was done by blue-green algae, and that they have some membranous extension of the plasma membrane in cytoplasm, which are known as chromatophore, and chromatophore, they contains photopigment which are in position to trap, that is light energy. Chromatophore, they are similar to the chloroplast of eukaryotic cell. Then we have to say cytoplasmic inclusions. Reserve food will be there in prokaryotic cell, definitely. Stored food is there in cytoplasm. That is in the form of, that is inclusion body. That is, they do not have any membrane here. They are free in cytoplasm. That example is phosphate granules, cyanophycin granules, or that is glycogen granules. These are the inclusions which are found in the form of stored food. In certain cases, as in case of blue-green algae, that is gas vacuole is also found, and these gas vacuoles found in blue-green algae and purple and green, that is photosynthetic bacteria, they help in, that is floating of this bacteria. Right? Come to the next now, that is nucleus. Many more times we have discussed, I told you that in bacteria a well-defined nucleus is absent. It is having this chromatin material, where no well-defined nucleus means here nuclear membrane is absent. Here in case of nucleus, that is nucleolus is also absent. In prokaryotic cell, nuclear membrane is absent, I told you. Genetic material basically is naked, that is, it is not, that is enclosed in a nuclear membrane. In addition to this is a very important point, that is here, you see, that is the histone protein is also absent, which is that is associated with DNA. This is absent here, and that is in addition to the genomic DNA. This is the genomic DNA. This one is the DNA found in nucleus. That is, many bacteria, they have a small circular stent outside the, that is genomic DNA, known as, these are called, that is plasmids. And plasmids, these are very important one. Plasmids will be of different type, and these plasmids, sometimes they help to develop resistance against, that is antibiotics. That is one character is known as, that is resistance to antibiotics. The plasmids are commonly used, that is bacterial transformation, and to transfer, that is DNA from one to other. That is, plasmids are frequently used nowadays in genetic, that is engineering work. Is it right? So that was about part first, in which we have seen, that is cell, the basic unit of life. and we have seen cell theory and detail of prokaryotic cell. Thank you.